Hello everyone, my name is Zhenghao Jin and I'm from Kunshan University. Today I'm gonna talk about bacterial transformation. The transformation contains three parts: the construction of gene expression vector, introduce the recombinant plasmid into the recipient cell, and the expression of the foreign DNA. The recombinant plasmid is literally what it means. It is a combination of the gene of interest and a large proportion of the original plasmid in E. coli. We get the plasmid from the E. coli cell and use the restriction enzyme to cut it and enabling the gene of interest to be connected with the plasmid. The sticky ends are unique base sequences that ensure the specific binding of the plasmid and the gene. DNA ligase is used to recombine the two parts together and make the recombinant plasmid. The recombinant plasmid we use in this experiment is PUC19. It contains the LAC-Z gene as a gene of interest and the AMP resistance gene as the marker gene to filter the cells that are not transported. Both the two genes are of irreplaceable significance because the antibiotic resistance gene can function to select the cells that are transported, and the gene of interest can express certain proteins. Let's move on to see how can plasmid get into E. coli cells. We use divalent calcium ions to make the E. coli cells competent cells, which means that they can take in foreign DNA. After foreign DNA is incorporated into its own genetic profile, proteins that foreign DNA encodes for can be expressed in these E. coli cells. Having a deeper look into the E. coli, only when the plasmids and the plasma membrane of the competent cell are close enough does the plasmid have opportunities to get into the cells. Also, heat shocks at 42 degrees is needed to improve the fluidity of the membrane to actually let the plasmids in. In this experiment, we use two kinds of mediums, and both of them contain LB, X-scale, and IPTG. Ampicillin is added to one kind of them. LB is the organic and inorganic nutrient that growth of bacteria needs. X-scale is a substrate of beta-galactosidase. Ampicillin is the antibiotic that functions as a funnel to kill the cells that are not imported with recombinant plasmid. And most importantly, the IPTG is an inducer to enable the LAC-Z gene to express correctly. We can see in the result that the group containing recombinant plasmids has blue colonies on the plate, since the X-scale is hydrolyzed by beta-galactosidase, which is expressed by the LAC-Z gene. The two groups on the plates which do not contain ampicillin have white colonies on them, and the plate which contains ampicillin but do not have the plasmids have no colonies. In this experiment, we can indicate that by taking advantage of the rapid reproduction of bacteria, we can express the protein we need in large quantities and reduce the cost at the same time. E. coli is a microbial of choice for expressing medical heterologous proteins, and this host currently produces approximately 30% of approved medical proteins. However, since bacteria are prokaryotes, they are unable to provide the post-translational modifications required for some proteins. Let's see how scientists solve this problem. When foreign genes are introduced into cow-fertilized egg cells, certain proteins can be expressed in the milk of mature cows, and these proteins are processed by the ER and Golgi apparatus. This specific way of producing proteins is called a bioreactor, meaning that we can produce proteins and get them from animals' external secretions. For example, we can produce human insulin using E. coli while produce human hemoglobin in red blood cells. Based on these findings, we are hoping that genetic engineering can function as a powerful tool in the future to benefit all people and even all creatures' lives on this beautiful planet. Lastly, I want to express my sincere thanks to DKU, Professor Lee, my dear classmates, and my family for supporting me during this learning process. And that is the end of my presentation. Thanks for watching.